This presentation is aimed at familiarizing students with the tools and equipment used in a chemical laboratory. A chemical laboratory is typically equipped with the following items: special types of glassware, fixtures to support the glassware, appliances to handle liquids and solids. Gases are handled in specialized equipment, which is not covered in this presentation. Devices for heating the glassware containing the chemicals used to create the chemical reactions. Precision instruments for the measurement of the mass of the chemicals used in the reactions. And precision instruments to analyze the products of the chemical reactions. Images of the basic items of glassware used in the laboratory are shown in the next few slides. The glassware that is used in processes requiring heat are made of Pyrex glass, a substance that will not crack under the stresses of heat and cold. These include beakers typically used to transfer liquids. Florence flasks are used to hold the starting reagents that will undergo a chemical reaction. When heating is required, a Florence flask is sometimes placed on a ring clamp to a ring stand and heated with a Bunsen burner. Erlenmeyer flasks are used to transfer liquids as well as to contain reagents used in chemical reactions. Boiling flasks are used in cases where a chemical reaction or purification process requires heating. For heating the reagents, a boiling flask is typically mounted in a heating mantle, a device which will be shown later. Two-neck boiling flasks have openings for other glassware such as funnels that are used to contain liquid reagents and a thermometer. The openings have ground glass joints which can be used to hold other glassware with ground glass joints such as a Liebig condenser. The three-neck boiling flask has openings for funnels, a Liebig condenser, and a thermometer. Other commonly used glassware include a Liebig condenser, which is used for distillation or for reflux when a chemical reaction is expected to take time. It has a cooling jacket that surrounds the tube leading to the flask. Cold running water enters through the bottom of the jacket and exits at the top. Reflux is a process that enables the reagents to be boiled for a long period of time, while the vapors of the liquid are continually condensed, and returned to the flask. Separatory funnels are used when it is necessary to separate two liquids that are not soluble in each other because of their different densities. The higher density liquid is drained through the valve, known as a stopcock, to separate the liquids. The funnel on the right of this diagram is also often used to add reagents to a boiling flask in order to create a chemical reaction. Test tubes are used to hold small volumes of liquids that are being tested with other reagents. A volumetric flask has markings to indicate that it holds a specific amount of a pure liquid or a liquid solution. Graduated cylinders are used to measure the volume of liquids in milliliters. A burette is used to add a precise volume of liquid or solution in milliliters to a flask for reaction or analysis. Glass tubing is used to set up special types of apparatus. The glass tubes can be bent and shaped using the heat from a Bunsen burner. Eyedroppers are used to add very small amounts of liquids to test tubes or flasks. For more precise addition of liquids, the pipette is used to draw in and transfer liquids to beakers or flasks. Another piece of glassware is the retort, a device used for distillation of liquids. It consists of a spherical vessel with a long downward pointing neck. The liquid to be distilled is placed in the vessel and heated. The neck acts as a condenser, allowing the vapors to condense and flow along the neck to a collection vessel placed underneath the tip of the neck. In the chemical industry, a retort is an airtight vessel in which substances are heated for a chemical reaction producing gaseous products that are collected for further processing. Such industrial-scale retorts are used in shale oil extraction, the production of charcoal, and in the recovery of mercury in gold mining processes. Now we look at some of the fixtures that are used to support the various pieces of glassware. Beaker tongs are used to carry a beaker containing a hot liquid. Crucibles are typically used to heat substances to a very high temperature. These tongs are used to remove the crucible from a hot oven and transfer it to a workbench or fume hood. Rings are used to hold beakers, boiling flasks, or Erlenmeyer flasks in preparation for heating with a Bunsen burner. The clamp is used to mount the ring on a ring stand and adjust it to the desirable height. Sometimes a square piece of wire gauze or a triangle is used to hold the flask or beaker. The burette clamp is used to hold a burette in the proper position on a ring stand. This image shows a ring stand with a burette clamp and a ring clamp to the stand. A wire gauze and triangle are also shown. The triangle has 3 ceramic rods mounted on wire in the shape of a triangle. Other glassware support fixtures include a holder to mount 4 funnels. The lab jack is used to raise and lower various pieces of glassware or other items to position them properly for the desired apparatus setup. A support clamp is used to hold various pieces of glassware to a ring stand. Other fixtures for glassware support include a test tube rack, a test tube holder, utility tongs, and rubber stoppers, which can be solid with no openings, or with 1 or 2 openings for the insertion of glass tubes connected to other glassware. Common items used in the laboratory for handling liquids and solids include a funnel as well as filter paper. Glass stirring rods. A Petri dish. This type of dish is usually used to culture cells for biology or biochemical experiments using agar, a gel-like material made out of red algae and different types of nutrients. Forceps for handling small items. A Buchner funnel used to filter solid precipitates from a liquid slurry. This type of funnel uses a flat, circular type of filter paper, and is usually attached to a flask with a vacuum connection in order to speed up the filtration process. The evaporation disc is used to allow the liquid part of a solution to evaporate in air, leaving behind the solid contents. Well plates are used for conducting multiple chemical tests or reactions using small quantities of reagents. The mortar and pestle is used to manually grind crystals or other solids into a fine powder. A crucible is a small ceramic cup used to heat solids to very high temperatures, sometimes to intentionally cause the material to decompose. The rotary evaporator is used to remove volatile compounds from a solution, using a vacuum to speed up the process. In this photo, the apparatus is set up to collect the volatile liquid that evaporates. Note the Liebig condenser that cools the vapors so they can be collected as a liquid in the flask at the left. Other liquid and solid handling items include the wood handle spatula, stainless steel spatulas, test tube brushes to thoroughly clean test tubes, polyethylene wash bottles primarily used for cleaning glassware. This slide shows some of the devices used to heat the reagents that are involved in chemical reactions. A boiling flask is typically placed in a heating mantle, which is plugged into a source of electrical power that controls the amount of heat applied to the flask and its contents. The Bunsen burner has been in use in laboratories since 1855 when it was invented by Robert Bunsen. The burner typically uses natural gas, which most laboratories have available from a valve on the workbench or in a fume hood. A multiple volume heating mantle can be used with a number of flask sizes and has the power supply built in. A hot plate with a magnetic stirrer capability is widely used in laboratories today. In addition to controlled heat, this device has the capability to rotate a Teflon-coated bar magnet that is placed in the flask or beaker. This enables the reagents to be stirred continuously during heating.
This photo shows some of the basic furniture used in a modern laboratory. The workbenches typically have a source of running water, and a large sink as well as valves for natural gas, running cold water, and sometimes vacuum from a central source. This photo shows a chemist working in front of a fume hood. Most chemical reactions are set up and conducted in fume hoods for health and safety. They provide a way for air containing noxious fumes to be drawn out and filtered before being released into the atmosphere. In addition, the thick piece of safety glass protects people from the dangers of spillages, fires, or explosions. Note that the chemist in this photo is protected by wearing safety glasses, gloves, and a laboratory coat. She is also holding the hot beaker with a pair of tongs. These diagrams show a typical setup for distillation of a liquid mixture using many of the items mentioned previously. On the left is a boiling flask fitted with a thermometer inserted into a one-hole rubber stopper. The flask is held to the ring stand at its neck by a burette clamp, and it rests on a wire gauze mounted on a ring. A Bunsen burner is used to heat the liquid. This boiling flask has a side neck that is attached to a Liebig condenser, which has two rubber hoses attached. The hose at the bottom is connected to a source of cold running water, while the hose at the top allows the water to run into a sink. The condenser is held on a second ring stand with a support clamp. As the liquid in the boiling flask evaporates, the vapors condense in the water-cooled tube, and the pure liquid, called the distillate, drips through an adapter into a flask or graduated cylinder. An example of a reflux setup is shown in this diagram. The reactants are placed in the round bottom boiling flask, which has a Liebig condenser attached. The flask is mounted in a heating mantle with controls for regulating the heat and the stirrer. A Teflon-coated bar magnet in the liquid at the bottom of the flask is used to keep the mixture stirred during heating. As heating takes place, the vapors from the boiling liquid condense in the cold tube above, and the liquid falls back into the flask. This enables the heating process to occur for a long period of time without the loss of any liquid. In addition to glassware and their supporting fixtures, modern laboratories are equipped with numerous precision instruments. This digital analytical balance is used to measure the precise mass of small amounts of solids in grams or milligrams. This two-pan balance was used for more than 100 years. It may still be seen in some older laboratories, but the digital analytical balance has largely replaced it. This type of digital balance is used to measure larger amounts of substances in grams, and is widely used today in research laboratories. The triple beam balance shown in this photo is still used in many schools, and can still be seen in some laboratories. Today's modern laboratories have numerous precision instruments for analysis. The atomic force microscope has come into wide use in recent years. It uses scanning probe microscopy, with resolution on the order of fractions of a nanometer, that is, less than 1/millionth of a meter. The information is gathered by feeling or touching the surface with a mechanical probe. This photo shows a modern optical microscope with a computer readout. The device has a built-in camera to enable images of the specimen to be displayed on the computer screen. Other modern instruments for analysis include the gas chromatograph coupled with a mass spectrometer, as shown in this photo. When a very small quantity of a solution is injected into a stream of gas and ionized in the chromatograph, the vapors of the ionized mixture pass through a column packed with a solid material. The vapors pass through the column at different rates because the lighter ions move faster than the heavier ones and are therefore separated. These ions are then passed directly into a mass spectrometer, which detects the masses of the atoms in the compound. The mass spectrometer thus gives a graph of the masses of atoms and groups of atoms in the compound, which can be used to determine its structure. Liquid chromatography uses a similar method to separate the compounds in a solution. In the high-performance, liquid chromatograph shown in this photo, the process is speeded up by the application of high pressure to the solution as it passes through the column. The compounds in the solution are separated, collected, and analyzed using other techniques. An important method of analysis is nuclear magnetic resonance, often called NMR. This photo shows an NMR installation. A sample of the compound to be analyzed is rotated in a magnetic field created by a large magnet as shown in the left part of the photo. While the sample is rotating, a wave of electromagnetic energy is applied to the sample. The changes in the shifts of the wave's frequency is measured and analyzed using the computer equipment shown on the right side of the photo. The graphical readout provides information used to determine the structure of the compound. Every compound has a unique interaction with light, and the technique of using light for analysis is called spectroscopy. The absorption of ultraviolet and visible light by a compound is measured using an ultraviolet visible spectrophotometer as shown in this photo. The signal produced in the instrument is fed into a computer, and the readout gives a spectrograph, which can then be compared with the graphs of other compounds for structural analysis. When infrared light shines on a compound, certain bonds stretch at specific wavelengths of the light. This infrared spectrophotometer provides a graph that shows the absorption of light at the different wavelengths where the various bonds stretch. For example, a carbon-nitrogen bond will stretch at a different wavelength than a carbon-oxygen bond. The spectrograph can then be used to provide important information on the compound's structure. Another instrument commonly found in a laboratory is the centrifuge. It is driven by a motor, which spins liquid samples at high speed. Centrifuges work by the sedimentation principle, where the centripetal acceleration of force is used to separate substances of greater and lesser density. Convection ovens are used to heat samples to specific temperatures, sometimes for long periods of time. Each compound has a unique melting point. This modern melting point apparatus has a thermometer immersed in the solid material. The analyst observes the sample through the lens, and notes the exact temperature at which the sample turns from solid to liquid. This value is then compared with that of a pure sample to determine its purity. This photo shows the old type of melting point setup that used a special tube filled with oil. The sample was packed into a capillary tube, and attached to the thermometer with a rubber band. The oil in the tube was then heated with the flame from a Bunsen burner. The analyst observed both the temperature and the sample to determine its melting point. The measurement of the hydrogen ion concentration is known as the pH of a solution. The pH is often measured with a digital pH meter, and a special electrode that is immersed in the solution, as shown in this photo. When a pH meter is not available, litmus paper is an inexpensive alternative, which can be used to get a close approximation to the pH of a solution. The future organic chemistry laboratory may be condensed down to just a few pieces of equipment. This chart shows a system for the continuous flow of chemicals to synthesize various pharmaceutical compounds. Today, pharmaceuticals are manufactured using batch processing at multiple locations. The disadvantage of this approach is long production times. In 2018, scientists and engineers at the Massachusetts Institute of Technology developed this compact, continuous flow synthesis system to produce a number of widely used pharmaceuticals. This presentation was produced as a public service to help middle school and high school science teachers develop experiments, presentations, and demonstrations that can be used in their classes. More science experiments and demonstrations are available on the TOPS of Santa Clara Valley website, www.topsofscv.org. TOPS, Teaching Opportunities for Partners in Science, is a program aimed at stimulating greater interest in science by middle school students.